Hello and welcome to today's webinar. My name is Emily Perry. I'm the Ensembl Outreach project leader. So I lead a small team that does various ways to tell people all about Ensembl. So one of the things we do is we deliver webinars like this. We also deliver live training. We produce online training and help and documentation. We answer your questions via a help desk, and we do our social media as well. So we're the people to ask for help if anything is going wrong. So my first sort of introduction, why do we need genome browsers? Well, we need genome browsers because genome data is big. The first genome to be sequenced was a bacteriophage back in 1977 by Fred Sanger, and it was a really big deal, and it was a really massive undertaking that this was done. The human genome sequence was finished, but as many of us know, it's still not finished, back in 2004, and that was 3 gigabases, and again, really big deal, but now we can produce genome sequences in a week, so we end up with massive amounts of raw sequencing data, which in itself doesn't mean anything. You have to work on this data, you have to process it in some way to make it into something that actually is useful. And genome browsers are kind of one of the ways that we go about doing this. So we're not the only genome browser out there. The sort of 3 major genome browsers are ourselves, but also UCSC and NTBI Map Viewer. People tend to have personal preferences onto which one they like to use the best, but it just largely seems to be personal preference. These are some of the features we have in Ensembl. So we have what we call gene builds for about 70 species. We take all of these genes, we compare them pairwise to one another to produce gene trees. We also produce a regulatory build which incorporates data from sources such as ENCODE. We have various ways to display variation data, and we have a tool for analysing your own variation data called the VEP. We allow you to display your own data in the browser. We also have data export via Biomart and programmatic access via our Perl and our REST APIs. The great thing about it all is that we are completely open source, so all of our code and all our software is free and open for you to use for whatever you like. We cover vertebrates in Ensembl. We try and cover a wide range of taxonomic space. We also try and cover biologically important species, such as model organisms, conservationally important species, and agricultural species. We have what we call a release cycle, which is why we're giving this webinar today. So in July, we released Ensembl 81. And we've now most recently released Ensembl 82. So our release cycle is around 2 to 3 months, and it involves incorporating in all of the new updates to the data and the software that we have. The benefit of doing it this way is that everything is traceable, so everything goes in and we bring out a new release all in one go. We put a release number on it so that you can use the particular releases, and you can go back in history and look in archive sites in order to get the older versions of data and match it up with things that you might have read in a paper or written in your own lab book. So let's talk about what's new in Ensembl 82. So one of the exciting things, and I'll go through all of these things in a bit more detail, is we have a new mobile site. So m.ensembl.org is where you can find our new mobile website. It's a bit stripped down. And we'll see a bit about what's in it in a moment, but do have a look and a play with that. We've got some nice updates to our software, so we've got a region highlight, we've got improved data upload, and we've also got some new data. So we've got some Zebrafish RNASeq data, we've got some an update to dbSNP, which includes data from EXAC, which is very exciting, and some more software updates to do with variation data. We've got a new table for looking at variants in genes, and we've got new plugins for the VEP in the browser. So when we're talking about sort of what we've got, there are different access scales for looking at data in Ensembl. So you can look at data, you can look at genes and variants one at a time, you can look at groups of genes and variants, or you can look at the whole genome's worth of data. So the main browser and the new mobile site are the 2 ways that you can really look at things one by one. If you're interested in looking at groups of data, you can use Biomart, the REST API, or the variant effect predictor to look at groups of data, but they're also able to look at things one by one. If you're interested in looking whole genome, the FTP site provides whole genome data, but you can also access whole genome data via our Perl API and our MySQL server, which are also capable of looking at groups and at data in a one-by-one fashion. So this is what our new mobile site looks like. If you're familiar with the main site of Ensembl, you'll see that this is a very much stripped down version of what Ensembl looks like. All we've got there is a search box. We've got a link to it. So we've got a search box in the middle. We've got a link to a species list, and we've got a link to some help, as well as some access to some social media, and also where you can email us about this mobile site. And of course, we can jump you straight to the desktop site from there as well. If you do a quick search for a gene, you'll see that you get the search results very similar to how you have them in the main site. And if we then look at the gene, we'll see we've got quite a stripped down version of what's there. We have these little pop-up, these arrows you can use to find menus for each kind of feature. So we have gene pages and we have variant pages. We don't have any of the sort of the larger things like Biomart or the VEP or anything working in the mobile site. And we think this will be really useful and interesting to people who are at conferences who perhaps want to quickly look up a gene that somebody's mentioned and they want to find quick information, or if you're at coffee and somebody mentions a gene or a variant of interest, you can quickly look it up. We do have, as I mentioned before, there is a link on the home page to access our email. So there's a quick contact there. You can tell us what you think about the mobile site, if there's anything you think it ought to have that it doesn't have, or anything if you're trying to use it, if there's any problem you have with it. Please do send us an email, have a play, see what happens, and the more people who try it out on different devices, the more we'll find out about any problems that there might be. So another feature that's new is the highlight feature. So this allows you to highlight a region of interest in the region view. This highlight remains as you move around or if you zoom in and out. And I think it's a really great feature if you found a favourite variant or an exon, something quite small when you're zoomed in on a region, then you can zoom out and you can still see where it was. Or you can move around the different regions of the gene and still see where it was, which I think is really useful. So if you have a region, if you're just looking at your favourite region of the genome, you can drag out a box, which you've always been able to do, but now you'll see that the pop-up menu that comes with a dragged out box offers to mark the region, which is the new thing, but also jump to the region, which is what it's always been able to do. If you do choose to mark the region, you now get this highlighting which appears quite subtle with this dotted line, but you can see this cross allows you to get rid of it, but also this highlighter icon also allows you to get rid of it as well. But it makes it much easier. I could now move around this region of interest. I could zoom right out and see an overview, but I would still know where my favourite exon or my favourite variant was in this region that I'm looking at. We've also updated our data upload form, so now it automatically selects the file type. It just makes it that little bit simpler. So if you're working with data on a URL, like I have done in this example, I paste in the URL of my file, it ends in .bam, and immediately this drop-down box selects BAM for me. If it's got it wrong, then I can change it. If I do an upload file, it also will automatically detect. The only difference is if you just paste in data. So if I were to paste in bed coordinates into this box, I would have to tell it that it was bed because it can't automatically detect it from looking at the data. It can only do it from your file extension. But it does simplify things ever so slightly. We've also got new RNA-seq data for Zebrafish. This is data that you can view in the region view. We've got RNA-seq transcript models that you can look at, and you can access them via the database. So if we go into the configure page menu from any region in Zebrafish, you'll see that you can go into RNA-seq and get a menu that looks like this. And you can see we've got a number of different tissues and developmental stages of zebrafish. And you can see that we've got gene models, intron spanning reads, and the BAM files for them as well. If I select everything for ovary and turn on the track star for these, you can then go into the browser and see that here I've got the RNA-seq models. So I've got a model of gene expression. I've got intron spanning reads, so you can see where these models came from, that these are reads that match up here and match up here, but have this kind of as a gap showing that this is that the read actually spans this whole massive intron. And we've also got the BAM, the alignments, the read coverage of the alignments themselves. So you can see where we've got these peaks. We've got matching up to this model as well, so you can see where all of this came from. So another kind of data that we have in Ensembl that's been updated in the new release is variation data. So we have two kinds of variation data in Ensembl. We have the small scale, which consists of one or a small number of nucleotides, things like small insertions and deletions, or single nucleotide polymorphisms. We've also got the larger scale variation types, so things like copy number variation, large scale deletions, insertions, duplications, translocations. And we classify variants depending on the genes they hit and what effect they have on those genes. So whether or not they fall within coding regions, if they change the coding sequence, if they fall in UTRs, splice sites, regulatory features, anything like that. And we assign them sequence ontology consequence terms. So this is standard nomenclature for describing variants. We also predict SIFT and PolyPhen scores on missense variants. So these are scores for changes in amino acid sequence, and they're based on how well conserved the protein is and also on the chemical change in the amino acid. So if you've got a highly conserved protein and you change part of it, that's considered to be dangerous because evolution's probably had a reason to keep it that way. And a chemical change is also, if there's a change from, say, a positive to a positive charge, that's not going to make a huge amount of difference, but a positive charge to a neutral may make a much larger difference. So that's the two things that are considered. And annoyingly, their scales are completely opposite to each other, but there are also predictions made on these scales. So in SIFT, if it's 1, then it's perfectly fine, and if it's 0, it's dangerous, whereas PolyPhen says that 1 is dangerous and 0 is perfectly fine. And everywhere you see these scores in Ensembl, you will also see the predictions alongside. We have variation data for a number of species, and also for a number of species in our sister site, Ensembl Genomes. And we have data from a number of different sources. And I'm going to zoom in on one of these sources, which is dbSNP. So in the previous version of Ensembl, we had dbSNP 142, which featured data from a number of sources, 2 of them being the International PatMap project and the 1000 Genomes project. When we've gone into Ensembl 82, we've updated our dbSNP to 144, and we've pulled in data from EXAC, which is a fantastic project. So this is a statement, a description from EXAC themselves of what they are. So they're a coalition of investigators seeking to aggregate and harmonize exome sequencing data from a variety of large-scale sequencing projects and to make summary data available for the wider scientific community. So this is a fantastic resource. They've pulled in these various large-scale exome sequencing data, some of which are disease-related, some of which are not, and they've pulled them all together into a single resource, which we've now been able to pull into Ensembl. And so we've got things like allele frequencies from EXAC can be accessed in some ways, but also all the variants that were identified by EXAC, often disease-related, because a lot of their data come from disease cohorts. For looking at these data, because we've got all these new variants, we've actually updated our variation table. Previously, it was a bit clunky, and there were problems in that we couldn't load the whole table of all the variants in a single gene in one go. And some exciting updates to the software mean that we now can view everything in one go. And we also have filtering options. So if you go into the filters, you can filter by things like variant consequences, but you won't have the danger which you had previously of it telling you that it couldn't display all the variants because of software limitations. You might be wanting to look at your own variation data, which you have in various different formats, and these are some of those formats illustrated here. And you might want to know what kind of consequences they have on Ensembl genes. And the way that you can do this is using the VEC. So there's a web interface for working with the VEC, and there's also a standalone Perl script. And one of the things that we've had available for the Perl script for quite a long time are VEC plugins. So these are extra functionality that we can add to the VEC. They can extend the output of the VEC, they can filter it, or they can manipulate it in some way. They make use of external data or code, so not necessarily data that's in Ensembl. It's also something to consider if they are making use of external code. If there's something, some kind of problem with that code, then that can have an effect on using them with the VEC. And they were previously only available if you were using the standalone Perl script, but we now have them in the web tool. So if you want to have a go with the variant effect predictor, you can find it on the Ensembl homepage. And if you go into the interface, you'll see that you can put in some data. So you can paste in data, you can upload files, or you can attach URLs of files containing variation data, which you can match against different sets of transcripts. And there are various options that you can then add to your VEC output, so you can choose what identifiers you wish to see. You can find out if any of the variants already exist in our database. You can get frequency data. You can get gene and protein structure data as well. Now, some of the things that are shown on this list are actually plugin extras. So things like CSN, unshifted HGBS, frequencies from EXAC, which was a project we mentioned earlier, microRNA structure and upstream-downstream distance are all extra features that have been added into the VEC by plugin. So if you were using the VEC script just a month ago, you wouldn't have been if you were using the VEC script a month ago, you would have been able to use these. If you were using the online tool, these would not have been available, but we've now made them available in the online tool. We've also got scores for protein changes, so we've got the SIFT and PolyPhen. We've got predictions of splicing events, and we've also got filtering based on things like frequency of the variants. But again, we've got some things in here that are new. So we already had SIFT and PolyPhen, but DBNSFP, Condel, and Loftool are all new have come in as plugins. The splicing predictions have all come in as plugins, and also conservation scores have come in as a plugin as well. So we've got this extra functionality added so you can work with your VEC data. You get, when you've finished doing all your input on the VEC, you should get a table that looks like this, or you can click through to get your results, and you get a nice big table giving you loads of information about all the variants that you've analysed that you can then work with to determine which variants you might want to study further and for what reason. So I've had a quick talk about some of the things that are new in Ensembl 82. There are loads more features that are in Ensembl overall, and we can come and teach you courses on these. So we can teach an Ensembl course at your institute for free, although we do expect you to pay the trainer's expenses. If you send me an email, if you're interested in this kind of thing, we can get sorting this out. We also, so we can do browser courses looking at the sort of point-and-click browser interface, or we can do API courses as well. If you're interested at all, this link here, and you should have access to the PDFs. So don't rush around trying to get this URL written down. You can read a bit more about our workshops, and you can contact me as well. There is absolutely loads of help and documentation, so we will be putting a copy of this webinar on our YouTube channel, where you'll find a bunch more animations taking you through features in the Ensembl browser. We also have we have online courses on EBI Train Online, which is how some of you might have gotten to this webinar. There are loads and loads of fascinating and useful courses on bioinformatics on EBI Train Online, including Ensembl courses. And if you want any help with anything Ensembl related, please send us an email to helpdesk@ensembl.org, and we can help you through anything that you're struggling with in Ensembl. Please do follow us. We have Facebook, we have a blog, we have Twitter, which again might be how some of you found out about this webinar and became registered. And we also have publications, and we like it if people perhaps cite our publications when they use our data in their research. This is the team who works on putting all these things together. So some of the things that you all have seen today. So the new export interface was done by Anne, who's here. The vet plugins were worked on by Will. I can't currently see where Will is in this picture. He's hiding from me. No, I don't know where he is. But he did them and he's brilliant. Various people have, the variation data was put together by Fiona's team, who I'm pointing to now. So all of these people did a lot of work on putting these together, and there's lots of going on behind the scenes that you can't necessarily see in the releases, but are general improvements on everything that's going on. All of these people have been working on. And this is just, again, listed their names and our funding sources as well, to whom we're very grateful for making our data available and allowing us to be able to do the things that we're doing. If you are interested in webinars, EBI do have a webinar series, and there's a number of webinars coming up until Christmas. So if you're interested in the complex portal, RNA Central, biocuration, Pride and proteome exchange, Uniprot, and then we'll be back again. We'll have a new release out in December, so we'll be giving a release webinar then as well.